Tonight, you'll be hearing from Dr. Jamie Matthews as well as myself, and we're going to be tag teaming a talk on mineral nutrition. So I'm going to go ahead and introduce Dr. Matthews to you all. Again, he's one of our research faculty in the department. He got his bachelor's in animal science from Rutgers University, and then his master's and PhD, both from Virginia Tech in animal science. So him and I shared that mutual experience of being Hokies there at Virginia Tech. He then went on and did a postdoc at the University of Florida's College of Medicine before joining the faculty at UK as an assistant professor back in 1998. He's had a very successful research career with numerous publications and awards, and some of those include both the institutional and individual National Institute of Health Awards for National Research Service Award, as well as the Midwest section of the American Society of Animal Science Outstanding Young Researcher Award. And then at the college level at UK, he's received the Distinguished Achievement in Agricultural Research, as well as Outstanding Service to graduate students. And most recently, and fitting for the topic of conversation tonight, he was awarded the American Society of Animal Science Cromwell Award for Research in Minerals, which is an award that's given out for researchers across multiple states that have established an excellent research program in mineral nutrition. And so with that, I'm going to turn it over to Dr. Matthews.
Okay. Well, thank you, and appreciate the opportunity to speak today. A number of you have probably heard me speak a number of times at the various beef bash and animal science get-togethers we've had with producer groups. So it's always a pleasure to be able to tell you a little bit about what we're doing and hopefully convince you that we're spending your money wisely and doing good things with that. So today I'm going to talk about selenium supplementation and particularly how that might be useful for if you're grazing or cattle that are grazing on endophyte infected tall fescue. Before I get started though, I often get questions from non-academics about what I do, and particularly during the summertime. And they say, well, now that classes are over, what do you do? Well, as this slide will show you, my primary research is 80%, and my graduate level instruction, teaching is about 15%, where I teach 2 graduate level courses, one in digestive physiology and transporters, i.e. how nutrients are absorbed across membranes, and then protein metabolism. So how do we metabolize primarily amino acids, and how does that interact with carbon and nitrogen pathways, et cetera. Also responsible for the graduate seminar. We consider this one of our most important classes. Every graduate student needs to take this once per degree. You can do good work, but if you can't tell someone about it, you're not doing very well. Important part, I think, of my role is mentoring a lot of undergraduate research projects. As you'll see, and I acknowledge to people who've worked on some of our data, a lot of undergraduates have done some really good things. The other thing we do a lot of is service. It's technically 5% of my appointment, but the peer review of other people's manuscripts and peer review of grants, departmental, college, university, industry, service, all really have a very synergistic effect on my research program. And that's what I'm going to talk to you about tonight's research. I work primarily with beef cattle. We have got a real nice project going with bovine immortalized kidney cell lines that express a lot of the transporters and enzymes that we'll be talking about today. So they make a good model to study and then ask, do these same things occur in the actual cattle? And then also I have a very nice relationship with Dr. Linderman for developing pig study, and this allows us to have our comparative physiology between monogastrics and ruminants. So it works very nicely. It's also very good for my students. So as most of you know who've listened to me speak before, I started off doing the research down in Princeton with selenium with Roy. Roy is our long-term extension professor, took a farm herd and changed it into a research herd, and also worked with Dr. Jim Bowling when he used to work with Roy about 40 years ago down in Princeton, and both Roy and Jim were responsible for all the grass tetany work and the mineral recommendation for the high magnesium that you guys all feed in late winter, beginning of the spring. That was all done with Roy and Jim. So we started working on the Selenium project together. They both have retired now. And after we got things going, we had fortunate opportunity to bring in Phil Bridges, who you heard speak about 3 weeks ago, I believe. He was talking about our various selenium forms and how they affect reproductive physiology. I'll be telling you about how to handle all the other metabolic stuff. And then I'm really excited to have Dr. Van Balen join us. Her expertise is in copper metabolism and nutrition, and I think it'll make a really good fit with what we've gone on down in Selenium and really extend that. So we're very pleased to have been able to hire her. And then again, Merlin Linderman, he helps with my stats, which is very, very important. You do what I do, as well as collaborates with the pigs. This is a partial listing. Some of the recent students have worked in my lab, who they are, what level of training they are at, and then where they've currently gone. So today's work, you're going to see work from Raquel Hagee, Darcy Mayman, Yang Jia, Ching Li, Brian Reisman, and Chloe DiGiorgio. They're very good students, and they've all done very well. Chloe's still with us, but the others have moved on to very nice positions. So it's very gratifying when they help us out and we can help them out. So what have we been working on? Most recently, I have two mainly funded projects from the federal government. One is the inter-organ glutamine metabolism, and basically this is some really basic work looking at how we can increase meat quality by enhancing glutathione and glutamine synthetase metabolism and capacity in finishing versus developing cattle. What I want to speak with you tonight about is this question of, can the form of supplemental selenium ameliorate fescue toxicosis? This current project is a compilation or a marriage, if you will, of two long-standing projects I've had. One was to answer the question of what's the optimal form of selenium that we should include in our vitamin mineral mixes for Kentucky and other cattle consuming selenium deficient forages. And as you can see by this map over here, Kentucky is in barely adequate or subadequate on the selenium, all these yellow states, and then all the red states are really low, very low in selenium. So it's not just a Kentucky problem. This research applies to basically 2/3 of the country. Ironically, these North Dakota and South Dakota, these plain states, they have their problem with selenium is that of toxicity. Their feedstuffs, their soy, or they don't grow a lot of soy, but the wheat, et cetera, they have about 10 times the amount of selenium that we have. So they face about 5-fold selenium toxicosis problems. So kind of interesting. And then the second part of this is understanding fescue toxicosis, and we've spent a lot of time in the last couple of years characterizing the biological mechanisms responsible for that. And as you know and can see from this map, Kentucky's right in the middle of the fescue belt. And as the climate continues to warm, this belt's going to be moving up. So there are going to be more of these northern states starting to experience fescue and then toxicosis when the cattle consume the endophyte infected fescue. So these two projects, as you'll see later, yielded data that allowed us to ask this question I'm working on now, and that is, can the form of supplemental selenium ameliorate some of this fescue toxicosis or the symptoms? Before we get started, I really want to thank you, the producers, and the taxpayers and the industry people who care very much what we do, that we cannot answer a question like this or begin to answer it without the resources that a land-grant university brings to it. For example, Les alluded to a couple weeks ago that we had developed 3 different herds down in Princeton besides our normal commercial herd and purebred herd. These are ones that are developed there solely on either inorganic selenium, organic supplemental selenium, or as you're going to see tonight, what happens when you mix the 2 of those. So each of those herds requires 35 to 40 cows, and these are multi-year, multi-generational commitments. A lot of resources that the college provides and the land grant system provides, that in addition to all the supply money that I have to bring in for various federal grants and industry grants, could not happen without a big infrastructure to handle that. And then when we combine that with Fescue and the problems, we have our endophyte free fields as well as our endophyte infected fields, and this too requires quite a bit of management and commitment, long-term commitment. So the combination of these 2 big sets of resources to answer this one question is only able to do this at the land-grant university, and that is funded from you guys by our federal grants program. So with that, I would like to switch now over more into the scientific part and ask this question: why supplement selenium? We just went over selenium is deficient in the soils of Kentucky, so therefore the forages and the other crops are going to be low. The requirement for cattle is 0.1 parts per million. This is probably a range. It's not enough, in my opinion, for when cattle are physiologically challenged. You'll see a little later why that may be the case. And for example, in our Princeton herd, beef herd, I've been measuring the selenium in the forages, the corn silage, and the soy hulls we buy from a local producer. And you can see all of this, the range from 0.02 to 0.08, 0.04 to 0.08, and 0.02 to 0.05 over the last, say, 8 years does not meet the 0.1 parts per million requirement. So consequently, we need to supplement selenium. Remember your high school chemistry a little bit? Selenium is a metalloid, and it's right in the same column as oxygen, sulfur, and then selenium. These are highly reactive bond states. There's a lot of unbound electrons there, and so they become very active. What becomes important then is selenium, whether we're feeding this in an inorganic form without any carbon source, or when it replaces sulfur in some of the amino acids. And amino acids are the molecules that make up the proteins. So proteins that have sulfur replaced for selenium, and we'll talk about how that happens a little later, become very reactive, and they become very, very important for maintaining antioxidant capacity throughout the body. One note here is selenium, unlike sodium or phosphorus, it is an essential trace element. So we're talking about parts per million on this in terms of supplementing it. So what are the dietary forms of selenium? We have to feed this. What are we going to feed? Well, the 2 are inorganic forms, or selenate or selenide are the most common ones, and they're fed because they're cheap, is the bottom line. They will meet your selenium requirement. By the end of this talk, we'll see that sometimes meeting that requirement with an inorganic form is probably adequate. Many times, however, when you physiologically challenge the animal, it's not. And so in that case, we need to supplement with organically bound forms, or at least the data suggests that might be a prudent move. So what are our different forms? When we say organic, what we're really talking about carbon. If you remember, this is carbon, these little junctures here, they're all carbon. So this is our quote unquote organic, is our selenium attached to a molecule that has a lot of carbon. In this case, if this selenium was sulfur, it would be methionine. Because the sulfur is in place of selenium, it's selenomethionine. And it's going to be incorporated into proteins, what we call the non-regulated pathway, and you'll see the importance of that in a minute. Selenomethionine is the primary form that is found in most plants. So our selenium inadequate forages are really, we don't have enough selenomethionine, and you'll see in a minute another organic form called selenocysteine. And these are incorporated, if you remember your codons, each codon, genetic code encodes for amino acids. These transfer RNAs are the molecules that bind, each of them bind a specific amino acid. In this case, the selenomethionine binds equally to its transfer RNA as methionine. And so this becomes a one-to-one competition. When we have proteins that are made up by methionine, selenomethionine that has replaced selenium in a random, or sulfur in a random way, these are what we call seleno-containing proteins. And these are different from what we call selenoproteins. Selenoproteins have the molecule selenocysteine. This is, if this selenium was replaced with sulfur, this would be the amino acid cysteine. And this is incorporated in what we call into proteins by the regulator pathway, because this tRNA is actually going to bind a different amino acid and make selenocysteine on it. So it has to be very specific for that. So the selenoproteins have this molecule, and this molecule, remember the selenium is highly reactive. So selenoproteins contain a selenocysteine site, which is responsible for a lot of the metabolism that goes into cells, particularly the antioxidant metabolism. So they're going to break down your oxygen-free radicals. This is why you've probably heard people who are in the business of producing milk, the dairy farmers. In Europe, a lot of work is done feeding organic selenium and inorganic so that you can drive the production of selenocysteine, so it'll go into the proteins and help with the antioxidant load, goes into the milk, and then it helps the human. So what? Do we have these different forms? Are they really making a difference? Well, inorganic selenium, typically sodium selenite, versus organically bound selenium, which I'm going to label as in blue color tonight, and the inorganic is red. This organically bound selenium, for example, Altex selenium-enriched yeast product called Seldplex, contains about 85% selenomethionine and about 8% selenocysteine, are two organic forms of selenium, plus about 5–6% of some other organic selenium derivatives. Don't have time to go over everything tonight, but when you compare or feed in your free choice mineral mixes or in your TMRs equal amounts of inorganic selenium versus the organic selenium, the result will be you'll have a greater assimilation of selenium, which is considered to be a positive thing, in the blood, red blood cells, the liver, the kidney, and the skeletal muscle. Again, in Europe, they've, particularly during the late 80s, early 90s, they fed a lot of organic selenium and all, trying to drive selenium levels up in milk and in muscle, skeletal muscle for sale, as a way of having sort of a nutraceutical positive effect by selenium. So there's some interest in this country of feeding different forms, and can we get more selenium in the meat, the muscle, and have a better form, making beef a more saleable product. Question becomes, from biochemical level, at least to someone like myself, why does this occur? Well, this obviously is a very complicated chart. I just want to pay attention to these circled highlights. All selenium, whether it's coming in an inorganic form, are coming in and being made in the organic form of Selmet, Selenomet, or Selenocysteine, eventually all end up as selenide. And selenide then is either excreted in the lungs or urine, different compounds, or it's going to be made, conjugated with phosphate and made into selenocysteine. So this is what we call part of our regulated pathway. So we're making this. The recycling of your inorganic forms to start with and your organic, by the time they come through everything and you get broken down to the inorganic selenium, all are going to go through this process and end up selenocysteine. So that's what we call a regulated. There's not much recycling going on there from your inorganic forms. In contrast, if the animal or ourselves are consuming organic selenomethionine, selenomethionine is treated like methionine by most of the enzymes. So it will constantly recycle that selenium as the selenomethionine molecule. So its recycling, what we call the half-life, is much longer. And so when we feed, we mentioned earlier, if you have inorganic versus organic, if you feed that and your blood levels go up, it's due to selenomethionine being in proteins in the blood, and they last longer because they just keep recycling, breaking it down to methionine and keeps recycling. Eventually, that'll get broken down and come into the selenium will end up as the inorganic form and either be excreted or, again, be put into selenocysteine. So with that in mind, what happens to this metabolism when we feed inorganic versus organic? So when you feed inorganic supplement, it gets into the gut of the cow, and it's going to come in as selenite or selenate. It's going to have a very short half-life, made into selenite, and then be made into selenocysteine, or it's going to be excreted. So when we measure the physiologic response, growth, gene expression, whatever, for inorganic, this is what we're driving, this pathway. When we feed Celplex or some other organic bound selenium source, which has the selenomethionine and the lesser extent selenocysteine, we're driving this pathway. So when we look at the effect of inorganic versus organic feeding in our free choice mineral mixes, we're really looking at driving this pathway or driving this pathway. So when we looked at this, we considered some of the consequences of this understanding. One, the dietary form matters. Selenium metabolism is a function of recycling. Proteins that contain selenium, the selenium as selenomethionine, have a longer term recycling. They last longer. The true selenoproteins, those that contain a selenocysteine, get turned over very quickly. So what happens? Why not mix this? Why not drive both these pathways at one time? And so that's the question that John Johns and Roy Burris and I were talking about when they were thinking about switching over the formulation in our UK beef IRM cow-calf mineral mix, whether we were going to stick with inorganic or do some organic or do some combination. So we started doing some testing to see what would happen. So we asked the question, what happens if we mix these forms? So basically then, without outlining the green, we're feeding, instead of 3 mg a day as inorganic or 3 mg as organic, we're splitting it. 1.5 mg is inorganic, 1.5 mg of organic, and driving both pathways at the same time. So instead of one or the other, we're driving them both. So to investigate this in our second experiment, we looked at this in slow maturing Angus cross heifers that were fed a total mixed ration. This way we could mix it in, and these animals are not physically challenged or physiologically challenged. Heifers growing about, maturing heifers at 1.1 pounds a day, they're not being stressed at all. In fact, we've obviously are limit feeding them to keep them at that growth rate. So we conducted this long study, 224-day supplementation study, where we're looking at either 3 mg a day as inorganic, organic, or mix. And then we asked what happened. Just to give you some of the data, this first graph shows you the whole blood selenium. And the black line here, shows you the effect of our control animals. Had no selenium supplementation, but they were borderline adequate or almost inadequate, if you will. The line that you can't quite see, but I've sort of circled with the red thing, this is what happens when you feed selenite. And then this is what happens when you feed the mix of selenite and Celplex, or just the organic form itself. And so you can see that these two forms that had any organic in them behave the same way, greater concentration of whole blood selenium than in the inorganic, and both of those groups were greater than the control. What about the liver, which is probably a better indicator of whole animal status? We see the same pattern essentially, where the inorganic is higher than a non-supplemented, and both the mix of inorganic and organic plus the organic, the blue line here, give you about the same response. So in both blood and liver, they're giving what we would expect. And the consequence of this, if you just use concentration in the tissue or concentration in the blood, half the organic form, which is more expensive, gives you the same result if you mix with the inorganic than if you did whole organic. The other point I want you to get from this table is the timing, because very important for us to be able to make recommendations that if one of these treatments becomes more beneficial than the other, when do we need to start feeding it before we see these responses? And this particular experiment is very typical of many we've done, where after probably about 28 days, or certainly by 56 days, the liver levels have stabilized. So this to us means that we can, if we're going to want the effect of one of these selenium forms, we want to be feeding about 60 days before we expect to get that effect. So one of our next questions was this difference we saw where mix equal organic selenium in yielding selenium concentration in liver, which is greater than inorganic, which is greater than control. Does that any way related to the pattern of gene expression? In other words, the proteins that are going to be expressed, and then the physiological capacity that comes with that protein. So to look at that, we did some transcriptomic analysis where we're measuring the amount of messenger RNA in the tissues as a response to, as a marker for gene expression. And what you can see here is that these are individual animals. These are individual genes for each of those animals that changed changed compared to the other treatments. First thing you can see is all the animals that were controlled were more similar to each other than organic, than the mixed animals or the inorganic animals. And this heat map, you look at the pattern of expression, the more blue it is, the lower expressed the gene, the more red it is, the more highly expressed. And you can just see, if you draw an imaginary line down here, keep drawing it down here, you can see the demarcation of how these clusters of genes respond differently from those treatments. So what it's saying is, when we looked at that slide of inorganic pathways-driven pathways versus organic driven, you're seeing that difference here now with specific genes and proteins that are involved with that. And these clusters, if we were to pull this up and look at all these individual gene names, we would see that these clusters represent groupings of proteins that function together and give you different physiological capacities. So what we're seeing, the significance of 8 clusters defined, is you have at least 8 metabolic pathways or physiological capacities that change in response to one of these different forms of supplemental selenium. So we conclude that yes, unique effects on gene expression pattern are achieved if you feed these different forms of selenium. This is in the liver. So if these selenium forms have differential effects, then on the message, then presumably the physiological capacity, what about the other big problem that we face as Kentucky cattle producers? That is the negative effects of consuming endophyte-infected tall fescue, what we have abbreviated here as E+. So the question is, what is the best form of selenium to include in beef cattle free choice mineral mixes to optimize E+ based cow-calf production systems? That's what we really want to know. That's what we want to know to make a recommendation to the producers, what should be in our beef IRM cow-calf mix. So I'm not going to show you all the experimentations, but as a summary from studies we've done, here are some genes that were affected by fescue toxicosis on these columns, and that were affected by the form of selenium supplementation. And these are about 4 of about 8 that we see. And you can see here that after 36 days of consuming endophyte-infected tall fescue, or 85, we get a major decrease in a protein called SEPase-HS1. This is involved in selenium metabolism, and what it means is you have decreased seleноcysteine synthesis. So if this gene is down, your ability to make all selenoproteins is going to be affected. Similarly, these other genes have other negative effects if you lose their, decrease their expression on glutamate, therefore nitrogen metabolism in the liver, glutamine, therefore nitrogen metabolism liver, and then importantly, one of the things that selenoproteins are known for, reducing oxidative stress, grazing endophyte infected tall fescue is going to decrease one of the major ones that's going to reduce their oxidative stress capacity. Now, because we're doing this as one project, mechanism of derivative effects of fescue toxicosis, at the same time we're doing a lot of gene expression work on the form of selenium, we matched up these things, came up some interesting findings. Inorganic and organic will both upregulate this SEPW, the same protein that fescue toxicosis, or grazing the tall fescue, decreases. Mix also will do that. They're all about the same. So this is really probably a function at not having enough selenium period in whatever form. But importantly, look how the other genes, how they were upregulated by the mix, not the organic, sole organic form, and not the inorganic form. And these have real major metabolic consequences. This is going to increase your selenium cysteine, therefore your selenoproteins, increase transport of glutamate, therefore your glutamine, your nitrogen metabolism and recycling in the liver. Same with this glutamine synthetase. And this upregulation of SEPW is going to decrease the oxidative stress. So these are positive results, obviously. So with that as our background, we designed an experiment where we're really asking the questions laid out in this little cartoon here. How do we go from taking these nice healthy cattle, they graze endophyte-infected tall fescue, consuming the ergot alkaloids, something happens on the molecular level and you get this. These animals obviously showing classic symptoms of fescue toxicosis where they urinate in the mud to relieve the heat stress. If you look closer here, you can see a concaving of your muscles where they're metabolizing protein and catabolizing, not synthesizing protein, etc. So we again want to take a transcriptome approach and look at what's going on that we achieve that. So this study combines then our endophyte studies with our selenium form. So we went back to our herds. Remember, as we began to talk, we have developed these 3 phenotypes of red, green, blue, and green being the combination where these herds are raised simply on and maintained on that. We took 8 steers from each of those groups, randomly selected from each group, kept them on their treatment, and then grazed them summer-long grazing, much like you would do on our tall fescue pasture. The only difference is from what you would do is that we did not have ad libitum intake we made sure they consumed their 3 mgs a day by mixing in a small amount of soy hull. So they consumed all of it every day. For our experimental studies, we need to do that. So we fed them, delivered the individual treatments in these kaolin gates. Many of you have seen these kaolin gates down in Princeton. You've seen the cows out in the pasture. Here's the transducers where each animal can go into one of these, get that. And we feed the form of selenium. Each one has its own treatment. So importantly, we're giving 85 grams a day, which is 3 mgs a day of selenium, which is 35 parts per million. If you're feeding, this happens to be Berkman's, if you were feeding Southern States, for example, they assume the animal's going to need 110 grams a day of vitamins or mineral mix. So their selenium, to get 3 mgs a day, it's going to be formulated at 27 parts per million. Importantly, none of these steers had any orts, meaning they ate it all, so we know the treatments were delivered. Many of you are aware that the hallmarks of fescue toxicosis is reduced serum prolactin, reduced serum alkaline phosphatase activity, reduced serum albumin. This is a function of the liver, nitrogen recycling and synthesis, and then the loss in average daily gain. Some of our work, just looking at the effects of fescue toxicosis on metabolism, showed would definitely affect the pituitary gene expression profile, alter the liver gene expression profile, and decreases glutamate synthase activity by the liver. So we are testing the hypothesis that the organic forms of either solely organic or one-to-one mix of organic and inorganic versus the inorganic would ameliorate, that is, block some of these negative effects or overcome them. So that's what we were testing in this experiment. So the first thing we want to look at was a whole blood selenium. And as you can see, these look similar to what I showed you just for the slow maturing heifers, where both the mix and the organic, sole organic form had higher levels of circulating blood than did the inorganic, which tells us our treatments worked, our model worked, we were delivering what we expected. Another major finding was that serum prolactin is decreased in when you consume endophyte infected tall fescue. That's a hallmark for it. So this is our inorganic steer group. And compared to them, our solely organic and our mix ended up having about 59 and 52% greater prolactin than we saw with our inorganic controls, which is saying that we have less of this prolactin suppression, so we are in fact ameliorating that problem. So the other ones I told you about, markers for selenium or fescue toxicosis, or alkaline phosphatase and decreased albumin. I'm not going to show you the data, but yes, we saw the same thing. So what about these gene expression profiles? The pituitary, and more specifically the anterior pituitary, is the organ that's responsible for hormones, secreting hormones and compounds that affect most of the negative responses we see with fescue toxicosis. When we see reduced milk, that is due to the prolactin being suppressed. We see Dr. Bridges talked about the negative effects of consuming fescue, endophyte infected fescue on the ovaries and the testes, and how he was also showing you how we can recover some of that by feeding the mix. But we also have the thyroid being affected here, and something we're looking at now, the adrenal glands, and how that's affecting the production of cortisol and therefore liver metabolism, as well as growth hormone being affected. So the anterior pituitary really affects most of the physiological symptoms we see associate with fescue toxicosis. So we conducted a transcriptome study, and here you see another heat map. This time the map is turned sideways. Here are your green steers, here are your blue steers, and here are your red steers, the inorganic, and they all grouped together except for one individual red steer. The gene pattern was more similar to the green and red versus all these other ones. But when you interpret this, you can see these are clusters of genes as outlined here, that major changes in the inorganic that are being turned on, these genes being turned on, or increased expression by the solely organic, and then by a mixture of your inorganic and your organic. You can see here how different this cluster of genes in your only organic versus your inorganic, or the mix of organic, and this cluster clearly being depressed actually by your mix. And that sometimes is a good thing, depends on the pathway. So we take all of this data, we put it in a bioinformatic pathway, and we establish the relationships between different metabolic pathways. That's what this map is showing. And it has identified the prolactin pathway with two other controllers, including thyroid releasing hormone pathway. So we take what's going on in a prolactin and we break that down and put it on the individual gene or protein level. And this would be in your lactotroph cells. And we're seeing that the organic form upregulated one of these receptors that is going to be involved in the production of prolactin. We see in the green form, we get a downregulation of a receptor, which results in an upregulation of prolactin messenger RNA. And then the other interpretation, when you follow these lines, is that the mixed form increases circulating prolactin by increasing the efficiency of the made prolactin in getting secreted into the blood. Whereas the solely organic form is increasing the efficiency of the transcription and the turning on more production of the prolactin. So they're doing the same thing, because both of these forms, remember, had increased prolactin in the blood, but they're doing it by different mechanisms. So again, not doing the same thing. What about the liver? This is a diagram showing your liver, just functional anatomy of the liver. Things differ. What I want to show here is the GS, which is decreased in endophyte consumption, and urea ornithine cycles tend to be increased because you're breaking down protein and getting rid of the nitrogen. So when we do a transcription factor study, what we're going to do is grind this whole organ and look at what's the effect of gene expression on all those pathways. So here is that heat map, looking at the red versus the mix, the combination of inorganic, organic, and then the organic. And again, you can clearly see these clusters of genes that differ. Here they're being turned off in the green as opposed to the blue and red mix. Here the red is mixed, these are up compared to both the organic forms, treatments with organic, turning them off. And here you have a turning on of both organic forms versus this. So very good, easy to see pathways being affected. When you take all this and look at just the nitrogen clusters, proteins involved in nitrogen metabolism, we see we get an increase in the GS protein in our mix, in our OSC versus our inorganic steers. And more importantly, the activity of these, when we do biochemical activity, we see that the organic, solely organic, has more GS activity than the inorganic, and the mix has the greatest of all. And this is very important. This says you're going to have a greater nitrogen recycling and efficiency in the liver. Of all those parameters, the one that we see that doesn't change statistically, quantitatively, but not statistically, is the average daily gain in these steers. Quantitatively, we get about, a 1/10 of a pound a day in the green and about 2/10 of a pound a day in the blue, but statistically, they do not differ. So what we're seeing in a weighing cattle after a 90-day grazing trial isn't enough to show the expression of what we're manifestation of all the physiological changes we're seeing. Now, when we do a test, a statistical test called post hoc analysis, and we ask this amount of response we got and the variation that occurred with 8 steers per treatment, if that same variation is true of the whole population of all steers, how many steers would we have for these to be significant? The answer is 40. So one of the things we're going to be doing in the future is conducting these same experiments, but just using 40 animals per treatment and seeing if these relationships hold. So overall, we see all these indices of fescue toxicosis being positively affected, ameliorated, if you will, by organic or the mixed form. We don't see the average daily gain. We completed a study this summer, or last summer, that was similar to this with non-UK steers, and we essentially get the same response. So we're very confident these are all real effects. So one of the things we're looking at was, we talked about the selenoproteins, and I mentioned the fact that they have this selenocysteine molecule, which is highly reactive. And I mentioned that they are really responsible for redox potential. That's what all these proteins are showing you. So this light blue, any one of these 25 selenoproteins that have the light blue are involved in redox reaction. So any of these that are upregulated are going to be positive and suggest that's going on. So we asked the question of the selenoproteins that bovines have, and we characterize that in both the pituitary and liver. We asked, are they affected by our form of selenium? And that's what this little cheat sheet shows you, that yes, the form of supplemental selenium differentially affects bovine selenium protein expression. Specifically, in the pituitary, organic forms versus inorganic upregulated these glutathione peroxidases and these other proteins involved in selenium metabolism. The mix upregulated a different group of proteins, selenoproteins. And so when we interpret that, both pituitary and the liver, we see that the organic form is giving us a greater capacity for redox potential, whereas the mix of inorganic and organic is giving us a better one for the oxidative damage that occurs. So again, making a real difference at the protein level. So with that, I think I've talked long enough, and Katie's probably getting a little anxious there. So I will be happy to stop and answer any questions that you might have. Thank you.
All right, so while we're switching out presentations here, feel free to type in any questions you might have in the chat.
Jamie, this is Dare. I've got a question for you. So, and this is, I guess, a little bit of a loaded question. It's obviously very important to you because of the, it's a great part of what you're working on. But is selenium probably the big one in Kentucky? I mean, I know we hear a lot about copper and all too. So you and Katie can fight that one out, but. Kind of in regard to Kentucky, how important is selenium in the big picture?
Well, I purposely did not get into the studies of looking at selenium deficient animals versus selenium sufficient, because we know it's important, and we assume people are following the recommendation that you're feeding at least a mineral mix that contains the selenite, which is the cheaper form. So our whole research has been based on looking at not selenium deficiency versus selenium adequacy, but in selenium adequate animals, what's the best form to face these, ward off, if you will, these greater challenges. So I'd say it's really important. In terms of our specific cattle herd down in Princeton, we tested for copper levels in these same animals, and we do not have a copper deficiency there in those steers that we use anyways. And I know that was the basis actually of Jim Bowling 35 years ago going down to Princeton when he was a researcher and working with Roy. That's why they had Jim down there. He was doing liver biopsies to look at the copper status because, as you know, fescue toxicosis, one of the side effects is to give you the light or orange colored hair, which is also common with copper deficiency. So
Yep.
I don't, Katie can talk, obviously speak to copper a lot better than I can.
No, I thought that was good, just to pointing out that it's not, yeah, the importance of looking at, you know, not deficient animals, but it's kind of more along the fine tuning lines of things. We know that deficient ones are going to suffer.
Right. Yeah. And to me, the big unspoken question, because I have a hard time getting a straight answer from the feed mills, is what's the difference in cost? And the best answer I got was probably from Berkman, and it's hard because they're at the county level, they're mixing up all kinds of different mixes for every county, seems to be different. A lot of that affects or is influenced by what antibiotic or fly control or what have you. But they would say using solely an organic form would add about $1 a bag to an $18 bag of mineral mix. Obviously, if you're using the mix, that would cost go down about half. But Katie and I were talking about this earlier. Neither one of us have a very good idea or can get a straight answer on that because it's kind of hard for the mills to answer because there's so much else going in those free choice mixes other than just the minerals and all.